You will hear about the microphysiological systems of the brain organoids. You will hear about the sensor technologies, and you will hear what it means to combine them with artificial intelligence. Because that's what organoid intelligence is about. The organoid is combined with AI.
Welcome to this Frontiers Forum deep dive session. I'm Lee Baker, the Editorial Adviser at Frontiers in Science, Frontiers' multidisciplinary open access flagship journal focused on transformational science for healthy lives on a healthy planet. Today's deep dive session is focused on the article titled Organoid Intelligence, a New Frontier in Biocomputing and Intelligence in a Dish. This is today's agenda. First of all, 5 of the article's authors will provide short presentations about aspects of the work. Lastly, we'll have a discussion about the ethical considerations with this research with Dr. Karen Rommelfanger. Our first speaker today is Professor Thomas Hartung. Professor Thomas Hartung is the director of the Center for the Alternatives for Animal Testing at Johns Hopkins University. He's made contributions to pharmacology, toxicology, immunology, and medical microbiology, as well as leading the development of the article we're going to discuss. Thomas also convened and chaired a community workshop on organoid intelligence, which formed the basis in part of the article and also led to the Baltimore Declaration towards organoid intelligence, which Thomas will discuss. So Thomas, welcome. Over to you.
Hello, everyone. It's a pleasure to introduce into something which was one of the most exciting developments in my scientific life. I have been fascinated by computers for long, and as long as we have computers, about 70 years now, we have tried to make computers more brain-like. Artificial intelligence is perhaps the most pertinent example of this. What we want to present today is how we can make actually brain cultures of human cells more computer-like. And this is what we call organoid intelligence, OI. We see that at the moment, AI is changing everything. And this is because of the synergy of increase in data. More than 60% is the annual increase in data in the world, which means that roughly 90% of all data have been produced in the last 2 to 3 years. Our computers, according to Moore's law, double their capacity over the last 50, 60 years, every 2 years. And AI, even more. 10 to 12 years now, we see AI doubling its capacity every 3 months, which means an 8-fold increase every year. And we see this in this enormous increase in capability, which is over these 60 years, about 1 billion-fold altogether. And this is why AI develops capabilities we could not dream of until recently. And this reminds me of this wonderful song of R.E.M. It's the end of the world as we know it, and very importantly, and I feel fine, because I believe that this is an actual advance here. At the same time, there's another disruptive technology which changes biomedical research, not as much visible for a general public, but very visible throughout biomedical research. While we had essentially no good access to human cells and human tissues, with very few exceptions of, let's say, blood and skin, since the advent of induced pluripotent stem cells in 2006, we have an ethical, clean opportunity to essentially produce any tissue, any cell type of the human body. And this enormous increase in methodologies, which led to organ-on-chip systems, which led to what we now summarize as microphysiological systems, is a revolutionary change because it is giving us highly relevant human organ systems. And this means, this is an article we were able to write in Science based on one of our workshops on the topic. This means new tools for drug development. And these microphysiological systems have in common that they reproduce the architecture and the functionality of organs. And for the brain, this means functionality, in the end, cognitive functions. And we are here in Berlin at the moment because we are organizing the second Microphysiological Systems World Summit, because this is a community forming which is at the moment trying to exploit for all of the different organ systems what is happening here. our own work on brain organoids started in 2016 when we showed how to mass produce standardized brain organoids. We were only the fourth group to produce brain organoids, but we were very impressed what you can see here in the background, these little flashes. These are actually neurons which are spontaneously depolarizing because they're in some type of communication. And when I presented this at AAAS in 2016, I very sloppy way said, they are thinking. And some people were shocked about the idea. Then I had a standard line which developed out of this. Yeah, but they have nothing to think about without input and output. And this was actually the starting point for what we are discussing now. These brain organoids, what happens if you give them input and output? So we are combining in this work actually 3 different types of technologies. And you will hear about these in the upcoming talks. You will hear about the microphysiological systems of the brain organoids. You will hear about the sensor technologies, and you will hear what it means to combine them with artificial intelligence. Because that's what organoid intelligence is about. The organoid is combined with AI. And we see that this has potential applications now for the physiology of learning, to have an experimental model to test what are the cellular prerequisites for these. It opens up opportunities for the testing of toxicants, which could fuel diseases, but also for the drug development process, where about 25% of all clinical trials are actually for brain diseases. And then there is the future of brain-machine interfaces, as well as possibly help us guide to understand how biological computing could be the role model for our computer architecture, or we might actually include some type of biological components into computer architecture, biological computing. So the big message here is we have finally a machinery experimentally available which is able for memory and learning, and we want to exploit this. And this is what our article describes, how these different components come together and how a 3D culture is enabling for the first time the basic mechanisms of a learning process. And there's an enormous advantage of moving from a 2-dimensional culture to a 3-dimensional, as you will hear from Lena in a minute, because we have higher cell density, which means more synapses. We have myelination because there's oligodendrocytes, which increase conductivity. And we have the glial cells necessary for long-term memory because they have to prune the synapses, the connections which are not being used. This is leading us to some ethical questions, like this article in Nature in 2020 already asked, can grown brains become conscious? I think the important question is, could there be some suffering? Would there be responsibility resulting from this? But there's also a couple of other questions which we want to address in this type of work. Is something we call embedded ethics, which means we work closely with ethicists. We design our experiments with ethicists. We are inviting them to our laboratories to show them what our experiments are actually about. What is very important for these ethical discussions is certainly, is the answer in the data, which means in our case, is the answer in the inputs this brain does receive. Can such a system experience pain if there's no nociceptors? Can such a system develop emotions if there is no emotions which are fed into the system? We are at this moment also thinking that this might actually be a possibility for experimental ethics and philosophy, because we can ask questions like, what is the minimum setup for intelligence to call something aware or possibly conscious? And an important part of the work at the moment, steered by Brett Kagan, talking later, is about terminologies. How can we apply and use the adequate terminology for these type of activities? And to some extent, we have to understand it is not just about modeling the human brain. We want to go, in some aspects, beyond physiology. There's no reason to limit us in size to the size of a human brain. Our brain organoids are not distracted by the many functions a human brain has in the human body. We can focus it on certain tasks. We are not bound to the embryogenesis process, for example. While oligodendrocytes show after about 5 months only in embryogenesis, we find them after 6 weeks in our cultures. We can also look into what would cells from a specifically gifted donor do. Let's think of an Asperger child. But we're also open to completely new architectures combining different components and units. We can add additional novel inputs, optogenetics and others. We can think unleashed in this case, because it is really a potential for synthetic biology, because we can also use interactions with reinforcement learning type of artificial intelligence, for example. So we laid out our ideas in a concept paper, which was accompanied with several articles. They're all available on the Frontiers in Science website. We were excited that several distinguished colleagues were commenting on our work, were giving us advice on how to develop this area further. You can find this on the respective websites. We also included a kit version and a laypeople version on organoid intelligence, summarizing our paper. Since then, we have received enormous interest. In the first 2 weeks already, we had about 500 press hits, including the Financial Times, the CNN, BBC, Forbes, and others who were reporting on our new concept of organoid intelligence. The important part is that we want to form a community now. And the Baltimore Declaration toward the exploitation of organoid intelligence and the formation of a community is key here. We have a strong team in Hopkins, but also very many colleagues, as shown today by Alysson Muotri and Brett Kagan, who became very integral parts of what we are trying to put together, which will show their own enormous contributions to this field. At this moment, the Whiting School of Engineering is making organoid intelligence one of the poster projects for a philanthropic campaign. So we are hoping that this will go viral and become something for which we are able to build capacities. As an instant response to our release, the White House made organoid intelligence already a bold biotechnology goal of the US. And in April, the National Science Foundation announced a program for engineering organoid intelligence, so an instant uptake in these ideas. We also created a new journal, Frontiers in Organoid Intelligence, as a sub-journal of Frontiers in Artificial Intelligence. I have the privilege to lead. And Lena Smirnova is the chief editor for this. And with this, I would like to close and hand over to Lena. This is really lived science fiction. And actually, this is a picture from 1966 in the Star Trek series, which I actually did grow up with, because Captain Kirk encountered here in 1966 a computer built on brains, which was actually biological computing, which at the time was pure science fiction. And now we try to make this a reality. Thanks a lot.
Wonderful. Thank you very much. Thanks, Thomas. That's a wonderful introduction and sets the scene for today's discussion. Okay, so on to our next speaker. It's Lena Smirnova, also at the Center, as Associate Professor at the Center for Alternatives for Animal Testing, also at Johns Hopkins University. Lena's work focuses on novel tests in developmental neurotoxicology, including the use of brain organoids, both to study learning and also memory and biological computing potentially. So Lena is, as Thomas mentioned, the chief editor of Frontiers in Organoid Intelligence, the new section within Frontiers in AI. Over to you, Lena.
Thank you, Lee. Hello, everyone, and I will take over and then build up what Thomas just recently said and talk about why we need new type of cultures and what we understand under the advancing 3D cultures like a brain organoid cultures. So when everybody who is not really associated with cell biology or cell-based technologies and methods would think cell culture means some cells which are growing on a petri dish, on a flask. And this is how we have been using that and doing that for many, many, many years in 20th century and beginning of 21st century. But if you look in the Petri dish, actually there is some limitations of those cultures, and actually the list is quite long. I will not go through all of them, but basically the monolayer cultures, we are not flat as the cultures, so they don't have a polarity, they have poor contact to cell-to-cell interaction, or poor survival usually, especially when we talk about brain cultures such as neurons. So there was a really push in the beginning of 2000, which is now ongoing, move away from these two-dimensional cultures towards more complex advanced models, which Thomas already mentioned, microphysiological systems. And so I highlighted here two, which is the organ on a chip and 3D cultures, whether it's spheroids or the organoids. Before I move to the organoid cultures, just briefly, what is organ on a chip? So it's not a potato chip. It's a small device, which is size of the USB stick, if you can imagine. And then scientists culturing different types of the cells, representing different organs, for example, lung, liver, kidney, on these small chips. And what is important, this chip is bioengineered device which allow functionality of this organ. So your lung on the chip will breathe, your heart on the chip will contract, and liver on a chip will metabolize. So that is one part of those microphysiological systems. So this is the umbrella term, and this word cloud show you what we understand under the microphysiological systems. And then, as Thomas already mentioned, we created a society, international society around this field to bring more developers and users and regulators together to move the field forward toward regulatory acceptance. So where we can use those microphysiological systems. So again, here you can see the example of an amylase chip, which was developed, one of the first ones which were developed for organ on chip technologies. So mainly, as for now, MPS are used for development of new drugs and efficacy and toxicity testing of those drugs, and toxicity testing of the chemicals. So this is the major fields where people are really interested. But you can also think about other industries like food, cosmetic, and tobacco, and so on, which are also using that. And here you can see also the list of different areas of the fields where you can think that it can really advance our work, make it cheaper, faster, and more predictive. For example, rare diseases, where not so many animal models for that, or unethical clinical trials, pediatric clinical trials, and so on, toxicity testing, countermeasures, which will be, for example, a big interest for Army, and so on. So there's big potential. And I must say that the biggest area who is taking up those technologies at pharma industry for preclinical testing. So back to the brain and the brain MPS or brain organoid. So you can see in this cartoon how versatile is these cultures, right? So when you imagine a brain organoid is a small ball of cells, and it has different cell types present there, which are important in your brain, such as different types of neurons, supporting cells, which support with nutrition, which provide isolation for the axons, or as a cord isolation for electrical activity, oligodendrocytes, neuroinflammation through the microglia, and so on. And with using brain organoids, you can study histoarchitecture of the brain, you can also study functionality, and you can study cell-to-cell interaction, for example, synaptogenesis, myelination, and neuroinflammation. And when you put those brain organoids in contact with the electrodes, which we called multi-electrode arrays, you can record the ultimate functionality of the brain, which is the electrical activity. And you can assess this electrical activity, depends on your system, whether it's disease or certain drug treatment. So as Thomas already mentioned, we have brain organoids developed at our lab, which we have, they're quite small. We keep them under 500 microns to make them very standardized. And you can see that we have different types of neurons there. We have oligodendrocytes present, which is, by the way, the cell, which is very difficult to recapitulate in vitro, but it's very important for brain functionality because it's provides myelination or like isolation cord, which I already mentioned, for electrical activity. And we can also have a good amount of astrocytes, which is, again, very important supporting cells for the neurons. And so our model is standardized because we're keeping them small, we're producing them in big quantities, in a big batches, so we can produce in one batch thousands of those organoids. They're reproducible from well to well, batch to batch, cell line to cell line. And as Thomas already mentioned also, that induced pluripotent stem cells allow us, which we generate them from, allow us to study different genetic backgrounds when we look into those brain organoids. And as Lee mentioned, what my focus is the neurodevelopmental disorders and developmental neurotoxicity, where mostly animals are used as a gold standard. Yeah, so if you want to assess how chemical, for example, or the drug affect developing brain, you will take a rat or mice and then run such an assays, which are very questionable about the ethics, is extremely expensive, time consuming, and we're dealing with a very complex endpoint. We're dealing with very low predictivity for humans. Therefore, experts in the field suggested that we can replace those animals with certain battery of tests, if you wish, where we can cover different key events of neural development: proliferation, apoptosis, neural differentiation, maturation, migration, and so on. And so far, this test battery it will be published in July as a guidance document from OECD, which is a huge step in the development, but it covers many different assays, many different cell lines, and mostly monolayer. So what brain organoids allows us to combine all those key events and analyze all those key events in one system, which again will reduce cost and help us to be faster. So in our lab, we use brain organoids for different aspects of neural development and neurotoxicity. So looking into the autism, for example, or gene-environmental interaction and neural developmental disorders, so how certain genetic predisposition may interplay with certain environments, exposure or lifestyle, and then which leads to the disease. We look into the cancer and so on. But what we haven't looked yet was whether we can study learning and memory on a dish with these models, or cognition. And then we think that we need to explore the capacity of our brain organoids, whether we can assess learning and memory. And so here you see again this chart of the certain aspects, key events of neural development, with the focus on network formation, synaptogenesis, and myelination, which is the key aspects for learning and memory. And that is our basic set for organoid intelligence. And as Thomas already mentioned, so I'm not going into details on this slide, these are prerequisites to have a successful model for learning and memory. It's active synapses, it's myelination, it's presence of supporting cells such as glia and microglia for pruning. And what Dolet is doing currently, so we are testing our current model without further modification of it, whether we can use some electrical or chemical stimulation and see whether we have a machinery of learning there. And you can see here that, for example, expression of early response genes NPAS4, which is a main player in synaptic plasticity, which is the prerequisite for learning, in response to certain chemical stimulation, suggesting that we have a synaptic plasticity in our model. The same is electrical activity. So you can see here, and you see more of network formation in brain organoids from Alysson later today, but you can see that we can modulate this network with different chemicals. For example, 4AP is seizogenic chemical, and you can see here the blue dots are the spikes. So every blue dot shows that one neuron is firing, and the lines shows that there are many different cells firing in the same time. So the electrical active at the same time, which shows synchronicity. And you can see that seizogenic chemical is inducing the activity. And if another chemical, which is another antagonist of the GABA receptor, also induces the activity. But if you block glutamate receptors, activity goes completely down. So we can modulate and show that our network formation is through those synaptic activity. We also stimulate them electrically, and as you can see, we can see red and orange. This is stimulation of one side of the chip of the electrode arrays, and we can record from a different side, showing again that the signal is propagating from one side to another. And so we use those organoids before we even think about biological computing. We will think about how we can use OI for the medicine, mainly for neurodevelopment and cognition disorders such as autism, intellectual disability, and neurodegeneration such as Alzheimer, multiple sclerosis, or dementia. And so we started this initiative we call DORI, and it is leading that Dementia Organoid Intelligence Research Initiative, where we can have organoids from healthy individuals and compare how they respond to stimuli of that's chemical, optical, or electrical, and how the Alzheimer patient-derived organoids responding to that, and whether we can compare and modulate that and develop their treatment against dementia or Alzheimer. And it's not only about Alzheimer, yeah. So we're collaborating a lot with our partners in the Hopkins and outside to develop such models for autism, for learning disabilities, and for some other rare diseases such as leukodystrophy and SYNGAP and cerebral disabilities. And you will hear more about our electrical activity building apps with David Gracias, who is the next speaker. Thank you very much.
Thanks, Lena. Our next speaker is Professor David Gracias. David has various appointments at Johns Hopkins, including the Whiting School of Engineering and the School of Medicine. His lab investigates micro and nanotechnologies, intelligent systems, and the interfaces between bioengineered and living systems. He has numerous patents relating to microchip integration, folding polyhedra, and integrated biosensors, among others. So over to you, David.
Hello, everyone. Thank you for coming to this webinar. Today, I'm going to tell you about new micro-instrumentation for brain organoids. So as you heard from Lena and Thomas, this is a new revolution in cell culture that we moved from planar Petri dish cell culture to 3D organoids. So just to put this in perspective, since some of you may never have seen an organoid, this is a little plastic dish, and each of those little specks in there is an organoid. And if you look at the scale bar, the typical organoids we've worked with are 0.5 millimeter. And so they are very small, they are alive, these cells are alive. And the important message today is that they are three-dimensional. So they resemble a sphere as opposed to a plane in 2D cell culture. So the way I think about this, and I've worked in microchip integration. If you open your computer, you have a motherboard, and it has different chips on it, the central processing unit and memory, etc., and a bunch of wiring. And it's a loose analogy, but we can think of the transistor being replaced by the neuron, the chip by these organoids. They may come in different sizes. They may be differentiated to do different things like memory, etc. And I think about how the computer, what will the motherboard, what will the platform look like? And the key challenge is, again, to remind everyone, these are alive. So they're fragile in a way. They need media, they need transport, or they will die. They are small, and they are three-dimensional. And one of the challenges in microchip integration and even in neuromorphic computing is that there are a lot of neuromorphic computers, but they are inherently two-dimensional or crossbar-based architectures. And here, for the first time, we have for free a 3D network, if you will, of processing units. So to summarize this, I've shown on this slide, you had the traditional conventional MEA plates. So if you look at them, they are two-dimensional. They are made by lithographic patterning, they're wiring, but they are inherently flat. And so if you put a ball on a flat plate, you can get signals only from neurites at the bottom of the plate. And so you lose a lot of information from all around the sphere. And so we, early on, we understood that we need some new kinds of instrumentation to get information from as many neurons or as many electrodes as possible. And, we have challenges with, again, they're alive, how to, whether we want to penetrate them. So there are technologies where people are trying to make bed of needles. Of course, if you penetrate the organoids, they can be invasive. So we took the approach of having a shell, and we model our device based on the human EEG cap. So if you look at an EEG cap, it surrounds the head. Why does it surround the head? And it's not a flat plate on the head, because it wants to record from all over the brain, which is three-dimensional. And we wanted to do it on chip so that in the future we could connect organoids to realize this dream of having like a differentiated organoids, interconnected with each other. So the main challenge, what we've done till date, and we recently published this last year in Science Advances, is that we made a shell for an organoid. And it's the big achievement here is in miniaturization. So the human brain is on the scale of 10 centimeters, and now we want to make a cap for a 0.5 millimeter object. And we want to make it in a way that does not kill the organoid and can surround and get data from all over the surface. We use what is called self-folding, and quite simply, it is a bilayer of polymers, and the polymers swell differentially in water or in media. So it's cell culture compatible. And importantly, as shown in the figure on the left in panel A, we can control what the radius of curvature, which is shown on the y-axis, versus by varying the thickness and the amount of cross-linking in the bilayer. And so we can make, if you will, just like baseball caps of different sizes, we can make a small, a medium, a large, and that is shown in panels B and C. B is the simulation and C is the realization. And so these organoids we envision in the future, they will come in different sizes. People are trying to make them bigger. And you can see the little gold dots there. Those are the electrodes. And so we could have wiring and electrodes in the shell. And for those of you who are electronics or micro integration people, we use planar lithography, just like is used at Intel or other chip manufacturers. So it's scalable, it's CAD mask designable, so you can tune it. You can add as many wires as you need, as many contact pads as you need. And these fold up, and we use a soft polymer contact, and our shell is also polymeric. And the nice thing is, the reason is that the brain is one of the softest organs, and people have already documented in the literature that you want to avoid a mismatch in modulus between what you're interfacing the brain with and the brain. So this is what we do. So we make the shells, and then we can do it on wafer. We have little container to hold the cells and the media, and then we can put it on a microscope and we record. So this is what it looks like. You can make many of them on chip. You can, in the foundries today at the chip manufacturers, they work on 16-inch wafers, 12-inch wafers. We work on smaller wafers. You can see the organoid is alive inside the shell. And so we demonstrated proof of concept that we can record using these organoids, and also we chemically stimulated them with glutamate, and we see an increase. So there are two real advantages of these 3D systems as compared to planar electrodes. One is the higher signal to noise. So if you have, as I mentioned, a plane and an organoid, you can record only from the bottom. And so when we took our organoid and we put it on a flat shell, we get signal that is far less than if we add the signal from all over the organoid. And this makes sense. If you're recording from all over the sphere, you add the signals, you will get more signal than what you would get if you were just at the bottom. And so the second idea, which we published earlier, is that, first of all, we can make them in variable sizes. So in the previous work, we made them down to few cell, and this study was done with IMEC in Belgium on wafer scale fabrication. This is a silicon-based system, so it can be made with different materials. And you can see here the spatio-temporal wave. So we envision that these organoids may have spatial waves of electrical activity that move around the organoid surface that can be recorded just like in an EEG system, and these may be important in organoid intelligence later on. So just to summarize, we've created a wafer scale approach for creating shells. They can be integrated on chip, they can be arrayed. We can use wafer scale processing to do the wiring on chip, off chip, and they enable, they're biocompatible, and essentially they move towards a more 3D platform, and maybe we can even map waves of activity in 3D around the organoid, which we anticipate will happen as the organoids similar to the brain. In the future, we are thinking of working towards integrating fluidics and also optical, like plasmonic, sensors in the shell to have a truly multifunctional shell. And important questions for the community are, what is the wiring going to look like? Remember, this is a liquid media. We might need an incubator, micro incubator systems. What is the motherboard going to look like? We have multi-core. And is there a Moore's law for OI, etc.? And with that, I'll end. And thank you.
Great. Thank you, David. Thank you very much. Next speaker is Alysson Muotri. Alysson is he's professor at the Department of Pediatrics, Cellular and Molecular Modeling at the University of California, San Diego. His work focuses on human brain development and evolution, particularly modeling the cellular and molecular mechanisms of diseases such as autism spectrum disorder in order to better identify and treat such conditions. Alysson, over to you.
Thank you, everyone, for attending. Thanking Frontiers to put this together. Thank Thomas for leading us on this new endeavor. My lab really started working on brain organoid as a model to understand how the human brain actually takes form. And this is because the human brain really acts inside the uterus, in the embryo. And that's a difficult experimental access for most of neuroscientists. We don't know when the first neuron fire. We don't know when the cortex network is formed. So there's lots of unknown questions because inside the uterus is a big black box. So the brain organoids provide us with an experimental tracking system to do that. So brain organoids are not a new technology. It's actually quite old. It was first developed by Yoshiki Sasai in Japan in 2008. And what he did was to neuralize human pluripotent stem cells in suspension, allowing them to self-organize and form what we now call a brain organoid. And by the time Yoshiki Sasai was able to demonstrate that the cells will migrate out and form really nice structures, three-dimensional structures that resemble the human fetal brain. And since then, many protocols have taken advantage of the initial Sasai protocol to advance or provide more incremental stimulations in different areas, in different protocols, and so does my lab. But before I continue on the protocol that we use, let me just pause and make sure that we are not talking about mini brains here. I think this audience probably understand that, but the media sometimes often use the term mini brain to describe those things and give you the wrong impression that you have a miniaturized brain in a dish. So these organoids have intrinsic limitations and differences compared to the human brain. First, they are immature neurons, meaning that they represent the early stages of the embryo, not an adult brain. They're not yet vascularized. We don't have all the cell types. So even the conditions that we grow these organoids are not optimized. So there's lots of things to do. And finally, I believe we're going to be discussing at the end, there's ethical and moral considerations as the science progresses. So this technology can move forward. So what we create is a semi-guided protocol. So we basically transiently stimulate the organoids with ectopic factors in the culture media to drive them towards the cortical fate or other brain regions and let the cell self-assemble. So we start with single pluripotent stem cells, and through a series of factors, we end up with an organoid that contains about 5 million cells in there in the size of a 0.5 centimeter in diameter. And during this time, there is a very dynamic process of cell populations that emerge during the organoid. I'm showing here a snapshot of a 4-month organoid showing that most of the pool of progenitor cells are producing glutamatergic neurons. At 4 months, we start to see the proliferation of astrocytes, so the shift between neurogenesis and gliogenesis is already happening. And we can see a pool of glutamatergic, GABAergic neurons or GABAergic markers that are not yet inhibitory neurons. If you give them enough time, they will become inhibitory neurons and will complement the network, will participate on the formation of complex networks. And that is an important factor for us studying diseases such as autism or schizophrenia, depression. These are diseases that do not affect the malformation of the brain, but they do affect how the networks connect to each other. So it was important to measure or to map this network in the organoids. And the way we do that is through the multi-electrode arrays or the MEAs. And you heard about this technology. We just place the three-dimensional brain organoid on top of the multi-electrode, and we record over time. The beauty of this technology is that it's non-invasive and allows you to measure the activity of the organoids over time. So we can actually do that through several years. And as we analyze the networks, we can see that the sophistication of these microcircuitries actually increase over time, providing us with a unique opportunity to follow the human neurodevelopment for the first time. So this is an overall brain activity over time, and I'm posting here some meta-analysis that we did with several groups using the MEAs, and you can see that a primate brain should fire about 20 hertz, and most of the in vitro systems that we have could not cross the 5 hertz threshold. So even organoids that were kept for 32 weeks could not cross this 5 hertz threshold. You can imagine the surprise that we had when with our protocol, we see an almost exponentially increase on the total firing hertz, hitting the 20 hertz threshold when the organoids are about 40 weeks or 9 months of age, which matches exactly the trajectory of the neurodevelopment. So since we have that much activity, we ask ourselves if these organoids were actually generating neural oscillations similar to what you can detect by EEG that pass through your skulls. And the reason why this is important is that we can bridge the basic biology, the study of the neuron synapses and molecular expression to human conditions. And this is what cognitive neurosciences have been doing for several years now. So long story short, we showed that organoids, they do not present these neural oscillations when they are about 2 months. It actually takes time. We start to see the oscillations when they are 4 months of age, very subtle. When they are 6 months, we see very stereotyped oscillations, and they become highly complex after 8 months, which matches with the emergency of several cell populations. For example, the increase in synaptogenesis happens at 4 months when we see the emergence of astrocytes. The complexity of the networks emerge when the inhibitory neurons actually become inhibitory neurons when they are about 6 to 9 months of age. So these allow us to understanding diseases such as autism spectrum disorders. And I'll give you like a quick example of how can we use organoids to actually model those conditions and move into clinical trials. So I'm focusing here on Pitt-Hopkins syndrome. This is caused by a mutation in a gene called TCF4, which is a transcription factor that causes all these clinical symptoms, which is considered one of the most profound types of autism. So when we create organoids from patients and controls and start comparing, we notice that the patient organoids are very small compared to the controls. And here you can see even the levels of TCF4 is about 50%, which suggests, again, that due to the mutations in one copy of the genes, we are producing only half of the levels of this transcription factor that is required for proper neurodevelopment. So the reason why these organoids are smaller is because they do not form neural rosettes, and they do not have as many cell proliferation as the control organoids. Here we are measuring the population of SOX2 positive cells. These are markers for neuroprogenitor cells. And you can see in blue here, where the patient-derived organoids, they have significantly less SOX2 positive cells compared to the controls. Because they do not have as many progenitor cells, they do not produce as many cortical neurons. We measure one of the sublayers of the cortex, such as CTP2 positive neurons, and we see again a dramatic reduction in the number of CTP2 positive cells in these organoids. By the way, this was all validated in post-mortem tissues. Unfortunately, one of the patients that we work with has passed away, and the family kindly donated their brains. So allow us to an orthogonal validation to confirm that the population of CTP2 positive cells was indeed reduced in one of these patients, which confirms the translatability of the human model system, especially compared to the mouse, where we don't see any profound symptoms that resemble the human condition. So we then decided to see if we can use gene therapy as a correction for this condition, where we clone the TCF4 gene upstream or downstream to a promoter that we created using the binding site of TCF4, which is called micro E5 boxes, to control the level of expression in individual cells. And our vector was able to self-adjust using a feedback molecular system to not provide any overexpression of the gene. So once we hit the organoids with the AAV carrying the cDNA of TCF4, we noticed that the number of progenitor cells increased, making the patient cells almost indistinguishable from the control cells. We could see also the rescue of SOX2 positive cells in the CTP2 positive neurons. So we are able to correct the cell populations that are affected. Moreover, we also measure the networks of these organoids, and we see that the networks on the patient organoids are dramatically reduced compared to controls. However, after the gene therapy approach, we're able to elevate the network close to the control levels. So finally, I mean, my lab has been using this strategy to model other forms of autism and test drugs or a gene therapy approach so we can move forward with clinical trials, especially because most of these conditions are human specific and you don't have any animal model that can represent it. We are launching now a gene therapy initiative at UCSD that leverage on these in vitro model systems. And I'll finish here by thanking members of my lab, but this is just a brief overview of everything that we do, and mention that I do have to disclosure that I'm a co-founder of Tismo. It's a company that uses brain organoid to find drugs for autism. Thank you.
Thanks so much, Alysson. Our next speaker is Brett Kagan. Brett is the Chief Scientific Officer at Cortical Labs in Melbourne, Australia, where he leads research building synthetic intelligence processors. You may well be aware of research that they published recently interfacing neural networks within computers in a simulation of the arcade game Pong, which he's going to touch on. So over to you, Brett.
Thank you very much, Lee. So pleasure to chat with you guys today, and thank you for the opportunity to really share some work with some of the other amazing researchers here who've given me an awesome grounding to progress forward. So essentially, you've heard of a number of different areas that people around the world are advancing. And what we're really interested in is taking many of those advances and trying to bring them together and combine them. And so you can break that down simply into a few different ways. For example, you can consider it as combining advances in hardware and software, neurocomputational understandings and synthetic biology, and bringing those together. For example, the improved hardware and software means that we can actually create what we call a real-time closed-loop implementation of an environment. More detailed theories or understandings how neural systems actually optimize behavior for intelligence. In our work recently, we explored the free energy principle, but of course, there's many other neurocomputational theories as well, mean that we can actually try and structure our environments in a certain way to motivate, and I use that term loosely, neurons or neural systems to behave in a certain way that we may consider useful. And of course, synthetic biology techniques which really underpin the generations of organoids, particularly the production of induced pluripotent stem cells, actually allows us to now have a scalable and specific neural cell production that is can be done in a fairly ethical way with very limited ethical barriers to achieve, which is very important as well. And so if you bring these together, you can say that then there's a number of requirements that this leads us to have. For example, we have to first grow the systems. We have to be able to generate neurons, and whether that's in monolayers or in organoids or something in between or different, it needs to be done in a way that is both specific, in other words, getting the type of neurons and supporting cells as well that you need, as well as in a scalable way. You need to be able to interface with them. And so I mentioned before the term a closed loop system. This is essentially where we can record from the cells, stimulate them, touch them on that in a second, read the activity, apply it to the environment, and then adjust to future neural stimulation, ideally in real time. This has to happen. And that's very cognizant for all of us. We all live in a real-time closed loop system because as we engage in the world, we are aware of the changes that happen to us as we move around and so forth. And then also we have to figure out how to program. And again, I use this term loosely. The information flows both to and from the neural system. How do you convey an environment, either simulated or real, to the neurons? And then how do you provide feedback about the neuron's activity in that environment? So in terms of growing, for example, some of the work we've done, we can make neurons through a number of neural systems, I should say, through a number of different approaches. Here's just showcasing some cells we generated that show markers of neurons, which is seen in neuron, beta-3 tubulin, which marks axons, and MAP2, which marks dendrites, as well as stuff like BRN1, which is a cortical marker supporting cells such as GFAP. And Ki67, there is none, because that's a marker of proliferation. And as many of you are probably aware, we don't want our neurons generally to be dividing, because if they do divide, quite often what they're turning is into something cancerous. And so if you can generate those cells, the next question is, can you interface with them? Can you design this structured information landscape to present the neurons to? And so that will require both input, which you'll have to present, as well as output. In our recent work, we looked at generating the game Pong. And so we actually provided here a topographical and place-coded way to represent where the ball was relative to the paddle. And then we read out from another area the activity of the neurons. And as the neural system altered this activity in a discrete, otherwise arbitrary to the structure of the neural system, as it changed activity in those areas, they actually were able to move the paddle around, and that changed their environment. And then finally, we had to figure out how we could program them. And I mentioned this term, the free energy principle for. It was a system that we decided to test because it had some actionable implications to it. In the simplest terms, what it suggests here is that a neural system tries to create a predictable environment, which means a non-predictable stimulation, such as something that's random, would be aversive, and a predictable stimulation would be reinforcing in terms of the patterns of activity inside neurons. And if so, the cells would try to modify their behavior. And so you can see here an example of how that looks. So neurons happening, showing activity here through columns. Hopefully it's a little clearer for everyone over there, but if not, you can go to SpikeStream, Cortical Labs SpikeStream, and look at a version of this at your leisure. But essentially what we're able to do is read this activity out and allow it to control the paddle and play the game. So we were able to look at learning in the neural systems. And so we can look at this over a number of different metrics, and there's a paper that's been published about this, but in short, we could look at the average rally length to see how the neurons got better at playing the game Pong. We can check a number of different control groups, and what we found is none of our control groups, which is here's a media-only control, which is CTL, IS is an in silico control, REST is when there's actual neurons there, but they're not actually receiving any information. None of these control groups showed any learning, but both mouse cortical cells, which is MCC, and human cortical cells, HCC, showed statistically significant improvements over time. And likewise, if we looked at different types of feedback, such as comparing the stimulus that I just described to you versus silent feedback versus no feedback, you can see here the stimulus feedback showed the greatest degree of learning, which was quite exciting to us to see that you can actually shape the performance inside these cells. Now, one of the things that we're really excited with is not so much this very, very early result. While it's nice to see statistically significant results that we can interact with these systems, of course, we shouldn't be too surprised to see that a neural system is able to learn. Neural systems are good at learning. It's why it's an interesting substrate to use. What I think is really interesting is now that we can start this iteration cycle of co-developing theories and tools, build the tool to test the theory, and then take the results to further refine the theory, then build more complicated tools to test more complicated theories. So this is a really exciting step forward. And I think what it will lead to, especially with the number of talented researchers both on the call and around the world working on it, is an exponential growth in the field. And so that's quite exciting. And so one of the things we've been doing to assist with that is actually trying to build out the tooling to support it. So here's a closed loop perfusion circuit. And I saw someone in the chat before was asking, how do you keep cells like this alive long term? This is one way that could be done. And it'll actually help perfuse the cells. And because we've got this system, we can actually keep cells on a playing a game now for extended periods of time. So here we're able to allow cells to play for about, I think it was 2 weeks in total, 18 hours a day of gameplay running on the system. And that was quite an interesting thing to be able to test, because previously we were limited to about 20 minutes. This works still in this early stage, but it does provide us with different opportunities. And especially for long-term maintenance, it's a powerful opportunity as well. And ultimately, I think a parallel can be drawn between, say, the nascent computer industry and the current, say, organoid intelligence at work that we're currently doing. So previously, go back 70, 80 years, you had things like an S360, it took up a room, you required a team of scientists in a lab to run. It wasn't very useful to sit at home. Now, one of the big breakthroughs with that actually was when the Apple II came out, and suddenly for a couple of $1,000s, anyone could have a PC in their home, they could program it, they could think of their own applications. And really, it was the community coming together and iterating and working on this sort of technology that led to the iPhone, or what we have these days, which is just huge potential in such a short space of time. And I think if you look over where we are right now, there I am very tired in the lab. That's currently where we're stuck at, this S360 version, right? It takes a team of people, it takes big incubators, it takes big freezers and all of this to keep these cells alive. But while groups and labs can make progress, that's going to be slow. The next step really is to generate a system such as a CL1, which is the next version of the perfusion circuit I just shown you, with an integrated MEA reader. And the goal for that is to make it as accessible as possible so that other labs and even other people can access it, either via the cloud or in person if you have a lab. And so ultimately, where does all of the research here lead us? Where are we right now and where we're going to go in the future? And I think it's fair to say that every researcher here would really highlight that uncertainty exists in so many directions where we're going to go forward. There's a lot of different challenges, but to tie it back to, one of the things Thomas said back in his talk at the start, there's links here to science fiction, and many people could look at this and be tempted to say, using in vitro brains for technology is science fiction. But science fiction ends and science begins once we start working on the problem and trying to solve it. And, I think what we've got here, which is really exciting, is a community-wide effort. And if we can work together to help each other to investigate this in a collaborative manner, I think we really maximize the probability for successful outcomes. And so I'll end on that. And thank you for being part of the community, and looking forward to the panel discussion now as well.
Thanks a lot, Brett. Thanks to all our participants so far, all our speakers so far. I'm pleased to welcome Dr. Karen Rommelfanger. Karen is the director and founder of the Institute of Neuroethics, and also an associate professor at the Department of Neurology, Psychiatry and Behavioral Science at Emory University. Karen has been involved for many years in the area of brain organoids. She served on the NIH Brain Neuroethics Working Group and also was an ambassador to the EU Human Brain Projects Advisory Board. First of all, before we start, research using organoids and especially brain organoids raises many ethical considerations. And Thomas touched on the embedded ethics approach within the Frontiers in Science article that's been led at Johns Hopkins. So from your perspective, could you begin by outlining for us the sort of major dimensions of ethics considerations as you see them?
This technology has the potential to impact each and every one of us at an individual and societal level. And with that come a number of questions we must collectively answer. And I want to bring up that term collectively because lots of you have importantly talked about community and the community we're building And we also should talk about who gets to speak in that community. So how might we as scientists, patients, citizens, policymakers, more proactively consider and then shape the trajectory of this technology to those impacts that will enable and empower us to have better lives rather than the alternatives? The discussions we had today are but one of a number of emerging brain technologies that have generated ethics discussions at the highest levels of government. The United Nations Human Rights Council is currently reviewing whether existing human rights law is even sufficient in light of technologies like these. And in 2019, the Transnational Policy Organization, the OECD, shared the first international standard in responsible innovation for neurotechnology. And this was adopted as a legal instrument by its 38 member countries. There are also questions as to how we might cooperate across the global enterprise of science. I see that we have people from all over the world on this participant list today. 5 years ago now, our neuroethics group, in collaboration with a consortium of the large-scale national level brain research initiatives across the globe, collaboratively generated the list of 5 key questions for scientists to consider in their work that took particular consideration to how different cultures might interpret the ethical and societal implications, and I'll highlight 2 of them. 1, what are the ethical standards of biological material collection, and how do these compare across global collaborators? So for example, should special regard be given to brain tissue and its donors due to the origin of the tissue and maybe even its past? And second, what is the moral significance of neural systems that are under development in research laboratories? Are the ethical standards for research conduct adequate and appropriate for evolving methodologies of brain models? So for example, if the model of organoid intelligence were in pain, would we be able to actually measure it? Given the rich experience and insights of these group of speakers, I'd like to use the next 10 minutes to ask questions to 3 of our speakers, and more discussion can follow with the whole group. And the first question is for you, Alysson. And what I have for you is around the idea of donor rights in the research context. One of the tools that we have towards ensuring research participants' agency and autonomy is the consent process. Participants and donors must be informed about the research in clear enough terms that they can make a decision about whether or not to participate. And we also saw one of the audience members, Kelly Johnston, asked about collecting samples from autistic donors who aren't interested in research that would cure autism. In addition, adequately informing participants about very sophisticated evolving science, such as with brain organoids, can be complicated. And as you noted, some preparations have developed similar electrical activity that resembles those of preterm neonates. So Alysson, I know you've been particularly mindful of this. Can you explain how emerging insights into brain organoids and their function has affected the way that you're handling the informed consent process?
Yeah, absolutely. I think a turning point was when we showed this sophisticated electrical activity that resemble the neonate human brain. So that changes everything, even our consent form. So our consent form now has to have very simple language explaining the capabilities of the organoids, including the possibility of reaching some kind of a level of a consciousness or even pain. So the patients are aware when they donate. We are not restricting the donation or the consent form to the specific signs, for example, treating Hopkins or treating autism, but these cells can be used for other things. We are sending brain organoids to the International Space Station to study microgravity, so they have to consent with all of that. We are sharing the cells with commercial collaborators that might make money from that, so they have to consent with that. So the consent becomes broader, but it has to contain like a language that a layperson can understand and decide if they want to donate the cells from their kids. Most of the diseases that we work on the severe side of the spectrum of autism. So the individuals, the autistic individuals, cannot consent by themselves. So the families have to consent for them. So there is a consent form for the families as well. So all of that is taken in consideration. So we were, in a way, not forced, but we decided to rewrite the entire consent form to contain all this language. And that's what we have right now. And we had, since we changed the consent form to incorporate all of that, we had just one family that refused to donate based on religion's purpose. But other than that, most people are still very enthusiastic with the science or the purpose of the science.
Thanks, Alysson. I think that's an important note for other researchers who might be a little hesitant to update certain consent forms for fear that they might, unintentionally scare away potential participants who would be really important to include in the study. Thank you, Alysson. So next, I'd like to turn to Brett and Thomas about the right to science. In 1966, the UN adopted the International Covenant on Economic, Social, and Cultural Rights. Article 15 of this multilateral treaty guarantees a right to benefit from scientific progress. One core feature of that right is that they be able to, as individuals and communities, have the information they need to engage in decision-making regarding research and development. We know that the more we try to model features of the human brain and the closer the approximation to human features, the trickier the conversation can become among stakeholders, including the scientists. Brett, I know this is an area you've thought deeply about, and even in your talk, you carefully qualified that you were using terms like motivate loosely. How should scientists proceed with respect to defining terms like intelligence and sentience, given their deeply ingrained cultural meanings, philosophical dimensions, and the public interest?
Yeah, it's an excellent question. And it's a very difficult one to explore, because to even explore the question requires discussion, discussion requires words. And if we can't agree on what those words are, it's hard to have that discussion. So I think the first thing, like I would say is most important, would be one, for people who use the language to try and be clear in how they're doing that. And I think given the interest we've seen in the recent time, when we started out, we tried to define the more ambiguous of our terms. And actually, I think what was nice was when we approached the OI, Frontiers was open and I think encouraging. Thomas and Lena were more heading that up, but of including a glossary of terms. And I think that's something more journals should adopt because it's very useful. Most journals don't allow you to do that, and that creates problems. The next thing that I think we need to do is actually identify the most problematic of these terms and actually establish a field guide. And with many of the, several of the researchers who are on here, and I hope some of the others will join us too once we move to the next stages, we are working on a nomenclature guideline to actually identify what these key features and these terms can be used and in what context. And I think context is also a very important thing, because if we talk about, let's say, organoid intelligence, like intelligence has over 71 distinct definitions. I think were caught about 10 years ago. Someone tried to characterize it, and there were 71 distinct definitions. There's probably more now. But if we talk about organoid intelligence, a reasonable person can identify that you're talking about something very basic and very simple. And so it needs to be understood in that context of where we're at the time. And I think so the challenge with that means that language will have to evolve as the field evolves. And so it will be a combination of people appreciating being made clear as we do the work as scientists to communicate and explain to the public where we're at the current time in our understanding. And I think that'll tie also in with what some of the other speakers mentioned in terms of actually identifying the neurocomputational metrics that might outline some of these. For example, intelligence or consciousness. To identify those metrics, we're going to need words to describe them. And as we understand and build better metrics, we can better understand and describe and define these terms. And so it has to be a constant iterative and evolutionary process as we move the field forward.
Great. I love that. I think that it definitely needs to be a evolving conversation where people are aware of the challenges of talking about these and the ease in which there is to misinterpret some of these terms. Thank you, Brett. Finally, I'd like to turn to you, Thomas. As one of the key visionaries of the field of organoid intelligence, expanding our suite of powerful tools now in non-human organoid or non-human intelligence models. And in the US, we recently saw a team testify before Congress about artificial intelligence, with a special focus on machine learning and large language learning models, wherein two of the speakers, including one of the creators of ChatGPT, raised concerns about the possible reality of artificial general intelligence. This spurred, of course, headline-grabbing media coverage, appealing to everyone's inner science fiction enthusiast, maybe even some of our Star Trek enthusiasts. And you also invoked R.E.M's lyrics in the opening. It's the end of the world as we know it, and I feel fine. So Thomas, in the current conversation about non-human intelligence, how do you see OI relating to conversations about the future roles, benefits, and risks of other non-human models brandishing the term intelligence, like AI?
I think we should first make very clear that we are at the very beginning and we are doing very humble experiments, actually. Many people think, when hearing about brain organoids and organoid intelligence, think we are talking to something which is in the lab. As you have seen in the variety of talks here, these are at the moment cell systems which have less neurons than a housefly. So we are really talking about something very small, and we are talking about the very basic first steps of learning and memory. The Pong game is certainly the most advanced of all functions which have been shown with these type of systems so far. So no danger of these systems taking over. Which does not mean we should not talk about it. And the first part of this is, and I think that's where we should learn from AI, is open access of information. I think there's never been a more democratizing technology than AI. As you have seen with some of the things you mentioned, like ChatGPT, 400 million people signed up in the first 2 months to ChatGPT. This is something which is, at least for small money, available to everyone. Many of these things are completely free. So I think the partnership of AI now with the world of biological learning, biological computing, is actually one which should follow the exact same path. And this is why community is so important. We are trying not to do the next Frontiers in Science paper secretly in our lab, like most people would pursue their research. We are trying to do this very openly. We lay out our plans. We show where we are. We show to everyone what the deficits are. We're trying to get funders like the National Science Foundation to roll out programs to make these things happen. So it's a very different approach to science, which I think we owe to the responsibility for such a possibly game-changing technology. And the fact that we are combining here several disruptive technologies, which are at the moment in an explosive way growing, namely the bioengineering, the sensor technologies, and AI in one, we might see some miracles happening in very short periods of time. So we are better prepared. And societally, being prepared means having a discourse, having many people involved, also using language which is as unambiguous as possible, and having a discourse on all levels, informing lay people, not coming with something which is a final product and then saying, buy it, take it, we have found something.
Thank you, Thomas. So Lee, I'll pass it back to you for the discussion.
Wonderful. Thank you so much, Karen. So yes, it's time now to open the floor to questions that we've been receiving in the chat. So we'll be going through as many of those as we can until the closure. So first of all, we have some questions relating to the cell cultures themselves, the organoid cultures themselves. One from Hyung Gyul Shin. Maybe these are to you, Lena. They might speak to better to your presentation, but please, others, you can join in. First question, what would the maximum size of brain organoids currently be, and what factors determine that? And an allied question will be, what are the main differences noticed during the differentiation between 2D and 3D? How do those differentiation processes differ when you translate to 3D with respect to the time to mature cells, the efficiency of differentiation, and so forth, and the functionality of the cells at the end? So size and differentiation characteristics. Lena, do you want to kick off there?
Yeah, I will start, and I will also hang over to Alysson. I think he would also comment on that question. So our model, which we developed, is relatively small, and it's on purpose. So we keep our model under 500 microns to avoid any necrotic core, because coming from the toxicology lab, main focus of our research was toxicity testing of chemicals, so drugs, and then so we need to have a healthy system from in to out. But other groups, like Alysson, for example, their organoids much bigger, so they let them grow 5 millimeters. Correct me, Alysson, if I'm wrong. So, and there is certain limitations with the size. Obviously, therefore, people are working hard on perfuse them. And I answered a couple of questions in the chat already about that. So we want to make our tissues healthy throughout the entire organoid. Therefore, Alysson and us and many other researchers are trying to make a vascularization, let's say, of those organoids to make them healthy also inside. Because the problem is the larger organoids, and it's well reported and confirmed that the core will be dying without nutrition in this type of size, in this size. How big we need them, that's a good question. And then this is what we're also trying to understand. So we are testing our current system, which is tiny, to see what is the roof. Yeah, what's our system, for example. So do we have a machinery there? Can we induce synaptic plasticity? Can we induce LTP, for example? And then increase in size and complexity will allow us probably to move forward if we get stuck in certain on the way, let's say, with the smaller organoids. Alysson, do you want to comment on the size, or I should move to the differences between 2D?
Yeah. The size, I think you said it all. I mean, different groups have different protocols. We can grow up to 0.5 centimeter. So it's very big. You can actually see a naked eye. And you are right. At one point, the center becomes necrotic because they're not vascularized. So all the nutrients are coming through diffusion. This is a serious problem in the field that many labs are trying to solve.
Yeah. So in terms of differentiation, I tried briefly to mention that in one of the slides. So the density of the cell cultures is very, very different. And we see immediately, even with a simple spheroids of one cell type, we saw already difference in how they react to toxicants, for example, in response to toxicants, or how we can keep them in culture. For example, we have a model of dopaminergic neurons, which we can keep in monolayer culture for maximum 10 days. And if we put them in 3D, so they're happily there sitting for weeks and months. So the lifespan and the time that that's one aspect. The complexity. So especially when we differentiate to different neural types. So mainly you get neurons, some astrocytes and monolayer. It's very hard to differentiate oligodendrocytes and monolayer, and even harder to have myelination in monolayer. So that's the three-dimensional cultures allow you to do that. And then what Alysson showed also, the histoarchitecture, yeah, the layering of the cortex, for example, yeah. So the structure of the brain organoids, you can do only in 3D, you can do it in monolayer.
Thank you to both. A couple of questions relating to the use of different organoid tissues in relation to brain organoids and organoid intelligence. First of all, from Angela Ramirez, in what other organs and organoids and tissues is OI being studied in conjunction with? And perhaps if I could editorialize a little bit there, maybe it might be worth touching on the use of sensory organoids as well, visual systems and so forth. So could we touch on the potential for the interaction between different types of organoids?
I mean, the human brain is a fantastic computer. It is hardly outperformed by real computers. And this is because it is not working by being fed zeros and ones. It is fed millions of different signals, which are also more or less analogous signals. And I think the brain can only shine if you give similar inputs. This is why combining it with sensory organs like retina organoids is a big way forward. And we have co-authors in our paper who are planning to do exactly this with us. And I'm aware of some work of Alysson's who is doing already these type of combinations. So it is, I think, a very important part. I mentioned nociceptors. I think this would be probably the simplest test type. And it could be a very important model for chronic pain to have nociceptors combined with a brain organoid. But we can also dream of completely different architecture. We don't need to have one brain, which is bigger and bigger with functional units. We could also combine a cortical brain organoid with a brain stem organoid. And David is already working on some models of combining units of brain organoids. And these are things which are all in the making. The limit is the sky here. And it can be, as I said, also hyper physiological. We are not bound to any boundary. We don't have to have it in a skull. We don't have to we can even give it new types of input we never thought about in biology.
David, do you have anything to add there?
Yeah, I completely agree. As I mentioned, I look at this as an analog to a microchip or a module in engineering. And I think there's a challenge. We need to learn how to build like USB cables, connectors between organoids, easy ways to plug and play, and then we can really make very complicated systems by connecting different organoids and building, organs on chip and organisms. So, yeah.
Another specific question that's come up with regard to applications in different tissues from Annie Moisan. Do you foresee applying a microphysiological system, an MPS, sensors, AI platform to model the gut-brain axis, i.e. two complex systems there? Is there any work underway or utility in that particular application for the gut-brain axis?
I can take this question. Annie, thank you. Great question. And we actually have been discussing that with one of our collaborators in US Army DoD department to do exactly that. It was a little bit before our OI concept came across, but we're really talking about having the chip where we will be connecting gut and brain organoids to do exactly, to study exactly that. So now with OI, it's a different level of complexity. We would be probably adding to that.
One aspect of the that was covered in the article in particular, which we haven't touched on that much today, is the data science approaches. We've touched on the bioculturing, we've touched on the bioengineering, we've touched on the sort of the aspects of intelligence. Brain organoids and the interfaces are going to create massive amounts of data and highly complex, noisy signaling. Thomas, is it possible, or anybody, to touch on the data science approaches which are outlined in the article, but what's necessary there to even make some progress and make sense of the communication here?
That's a very important point. We have to talk about the aspect here experimentally, because this is first of all really generating massive amounts of data. We are trying essentially to record about 1/10 of all of the neurons in the system, which requires a proximity to certain to these neurons. We are using not only shell electrodes, which are limited, like the EEG is very limited to how many contact points you can actually get, but we are using also Schenck electrodes. And the latest generation of these, which is not yet commercially available, has, I think, about 5,000 programmable electrodes in about 1 millimeter. And so we're really talking about highly dense electrode systems. There's also other types of connections to the system. So this requires really a big data infrastructure. And we are thinking at the moment, how can we work like the people have done in particle acceleration work in CERN, which is long times there's no signal, and then suddenly something is happening. How can we just store the events so that we only analyze the events later and don't analyze all the pauses between these signals? It will also then be, I think, extremely interesting as soon as we reach some capacity. The brain is an incredible computer. The capacity of the human brain is estimated at 1 exaflop. And 1 exaflop was exactly what was first surpassed by the largest supercomputer in the world 1 year ago, in June of 2022. And this was a 680 square meter installation for $600 million in Kentucky, the Frontier computer, which is the supercomputer at the moment. Yeah, this shows you how much the brain is doing, and also, let's say, why we have very, very careful hopes that some of this can be translated into neuromorphic computing that can teach us how to make more efficient infrastructures and get some of the unique characteristics of the brain computer into actual computers.
Wonderful. Well, on that note, looking to the future, a complex future, enormous challenges ahead. But perhaps we've discussed some enormous promise as well today. I'm afraid that brings the session to a close today. Thanks to all the presenters, all the speakers, to Thomas, to Lena, to David, Alysson, Brett, and to Karen. The Organoid Intelligence article we've talked about is available at the Frontiers in Science website. Together with the article hub I mentioned of content, there's an editorial by Rémi Quiron, the Chief Scientist of Quebec and President of the International Network for Government Science Advice. There are viewpoints by Carl Friston, Gary Miller, Arte Aluwalia, and a policy outlook by Julian Kindelöhler, who was previously the president of the European Group on Ethics in Science and New Technologies. You can continue the conversation on social media, #FrontiersForum. Please subscribe to the Frontiers in Science newsletter at Frontiers in Science website. So for now, once again, thank you for attending, and we'll see you next time. Bye-bye.